This is Young Onset Parkinson's Network. We believe young onset Parkinson's disease does not define you. You define it. We are a safe place to learn, connect, and engage within a supportive community that actually gets it, because we've got it too. Live your best life and thrive far beyond diagnosis. Welcome to Young Onset Parkinson's Network. Living well starts here.
Welcome, everybody, to the newest episode of the YOPN PMD Alliance Living Well Starts Here podcast. I am in Washington, D.C. with our friend Simon Allard from Amneal Pharmaceuticals. We are here for ATMRD, Advanced Therapeutics and Movement-Related Disorders, a Parkinson's Research Conference that I was kind enough to be invited here to attend and represent the patient voice. We're here, as always, with Chris, and unfortunately, Mel was unable to join us because of some technical difficulties, but we have her permission to power through and have Simon give us a little bit of peek behind the curtain of the pharmaceutical industry. And I'm going to turn it over to him to give us a little bit of background as to how he ended up in pharma.
Well, Hi, and thank you for having me. It's a privilege, really, to be part of your podcast. So I'm a scientist. I always loved science. My story is I watched the Hulk as a kid. And my favorite part about the Hulk was the part that he's a biologist and giving these cells and living entities new properties, doing research. And I didn't really care about what came next and the anger management issues that he has. So I focused on the good part, and I always wanted to be a biologist. So I did an undergrad in biology. I then took a class in the circuitry of the human brain just to try it. And there was a lot of detective work associated with learning the circuit, because a lot of how we learn how the brain works is when it stops working somewhere. So when there's a lesion in the brain, someone has a stroke, and then there's a manifestation, then you know that you have strong evidence that the region is involved in a particular function. And that's a lot of detective work that I think is very interesting in science. So I did anatomy and cell biology as an undergrad. And then I started studying pharmacology program master. And I did a PhD in pharmacology and neuroscience. At the time, neuroscience was not as defined as a field of its own, at least in McGill University, where I studied. So we had an associate program. So people that were already part of a program could elect to be part of neuroscience as well. So I'm a neuroscientist and a pharmacologist. I've always been very passionate about what I do. In fact, I prioritize my career a lot through my life. No one does academia for money. I say that I gave it my 20s and my 30s, and I really enjoyed the research. It takes a lot of passion. It's a lot of hard work. I was successful enough to survive, but not successful enough to be comfortable. I had to bring in my own money. So, I was at McGill, then I got a job at Hopkins, first for postdoc number 2. And then I became a research associate, which is the entry-level faculty, where you can apply for grants, get money, you're paid out of your grants, and your grant money is used to pay for your experiments and pay for the students that you're training as well. I was successful good enough again to survive, but then it was a lot. The passion takes you so far. I got tired a little bit, and I don't regret any of it. It was rich in many ways, but when I hit 40, I realized that the precarious life that I was living, where my job was not guaranteed, I had no safety, my income was guaranteed, and I had no savings because I was paycheck to paycheck. I was trying to find a way to still work as a scientist, because a lot of PhDs end up working for finance or something else. I really wanted to maintain that excitement and all that time investment that I put into becoming a scientist. I wanted it to be useful for something. I came into the lab one morning and I opened my Hopkins email, and I had this strange message describing a transition opportunity as a medical science liaison. And I didn't really know what that was, and I thought it was a scam, to be honest. And I looked it up, and the company was real, and the position existed, and I interviewed for it, and it turns out it's a job I didn't know of that turns out is a job that I really enjoy, which is a medical science liaison. where I wear many hats. I'm responsible for educating clinicians on our new data. I'm responsible for finding collaborators for clinical research. And because Amneal is a smaller company, then I also do some data analysis. We get feedback from the field from clinicians, and then I get access to the data, and I'm like, well, they're having this anecdotal observation. Can we see it in the data? Or they're having these limitations. Can I have something informative? I feel like I'm hired as a scientist, and it feels great. And I really
Awesome.
Yeah, so I mostly liaison between the science and the clinician. So the typical person I interact with is a neurologist. And
Gotcha.
so the neurologists, they don't have time to update themselves on all the new data, all the new publications. Our company, we publish data, whether through conferences, presentations, and through peer-reviewed publications. And I go around on my territory and I update the neurologist on like, okay, so we did this new analysis. This is what it means. So I'm there to present the data, save them some time, but also have a dynamic conversation. So they might give me feedback. They might have concerns about the data that I can disambiguate. They might have questions about the interpretation, the scope of the data. So that's more what my role is about. But, sometimes I'm privileged enough to have opportunities like this where I'm going to face real people. So I do have a lot of interest in that, and it's equally important.
So I like what you said about part of your role is to bring the data and get feedback back from the neurologist about what they're seeing in their practice. So I think a lot of us feel like when we go to our doctor's office that there's a gap between like what they have read about and case studies and what they're seeing, what we're seeing, the medication, it acting, in our body, And so that was a question I always had, like, how does that information get back to the company that's making the medication? And you're saying that's part of what you do.
That's a big part of my job. The difficulties we face, it's really in fostering or creating new relationships. There's some institutions where it's easier. There's some institutions where there's a lot of roadblocks. We face a lot of, and I'm not going to say that pharma is perfect, but sometimes some clinicians might understand if they're more research forward, they tend to interact with us a little bit more. But sometimes it's ambiguous whether or not we're commercial or so just to make it clear, I'm part of the medical affairs department. I'm not part of the commercial department. There's a separation of church and state that is necessary so that I maintain some level of credibility, that when I discuss data, it has to do with me as a scientist using my skill set to help them. Different institutions, different clinicians, we have different levels of relationships. Sometimes it's great. We have bilateral communication. We get feedback from them. And that's really how we keep it as a live document, a live analysis. And a drug, it's not over after FDA approval because phase 3s or phase 2, 1, 2, 3s are all experiments. For example, early onset Parkinson's patients are often not included in clinical trials because they want a homogeneous population, but a strong limitation. And then we need to be in the field out there getting feedback from the real world and have these discussions. Like you have patient types that were not part of our clinical trial that are you observing that they're having different responses? Is it better? Is it bad? How can we figure it out? So it really depends on the quality and the strength of the relationship. So then again, there's heterogeneity in a patient, depending on the clinician they see. If they are in a center that has a good relationship with the pharmaceutical, then there's more transmission of that information.
So, like I said, I've been at HTMRD for like 4 days now, and I've been talking to so many researchers, neurologists, and everything else. And this is something that I found out that crazy blew my mind. So I knew that there was some doctors' offices that did not let reps come to them. I said I go to Mayo. Mayo doesn't let pharmaceutical reps in their space. And so when I wanted to get on Crexont, I went to a general neurologist so I could get free samples. And, this is kind of my takeaway is what I've learned. It's like how I saw pharma sales, and I understand that you're not in that
Mhm.
division, he's in a different division, but how pharma sales works is that they're also going in and educating when they, as part of their role too. And so whenever we wonder like why our doctor doesn't have this medication or this treatment, like, Chris, you're on the pump, the new Anapco pump. I don't know that we know anybody that's on that. But when I was asking them like, why is that? Why do some of us have access to some things and others don't? Because you guys know I'm all about access.
Mhm.
And that is why they literally said, we don't have access. And I'm like, I know we don't have access. But to me, access means, can I get the medication or the treatment that I want? To them, access means the ability to actually be in the doctor's offices to give them that sample or educate them on the pump or whatever that is. So they're having their own access problems. And that blew my mind because we're all trying to do the same thing, which is get us the drugs and therapies that we need. But there's barriers on everybody, you know? It's just different difficulties. And then we wonder like why we have these problems, you know? And my takeaway is, that we have to then go back to our doctors. I was having this conversation like 2 weeks ago in my support group. Like, we have to feel empowered as patients to educate ourselves and ask our doctors to get that education that they need so that we can get what I need. So we always talk about advocating for ourselves, right? And but they have to do their part too. So we're kind of bidirectionally educating them in that same way. And part of that is that they should reach out and work with pharma. Otherwise, we're going to all keep being on all this different combination of stuff. And then as newer things are coming out, like stuff is coming all the time, that is going to be even more important that we all know about it and make sure that our doctors work with pharma to get it to us. It's just how the system works. And so that was like a real eye-opener
Mhm.
for me.
Yeah, I think there's understandably a trust issue sometimes between clinicians and pharmacies. There have been historically, you know, problems with abuse or pressure or corruption. And, but, we shouldn't all be painted in the same way. And I think it's our task from I guess, actionable item is to try to be as ethical and transparent as possible to really build that trust with the clinician.
Do you mind if you just briefly go over what the stages of clinical trials? Because we
Yeah.
always talk about, and I'm guilty of it myself. I'm in these rooms and I'm listening to them talk about, because I just want to know when I'm going to get it. Like, that's all I care about. And I know we all feel like, when are we going to get this stuff? So maybe if you could just briefly explain, like, what the phases are, how long they take,
Yeah.
and why it takes so long for
Yeah.
us to get treatment. Because I've told you guys, I knew about Crexont like right after I got diagnosed, and when it was still IP, blah 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 blah, and it still was almost 2 years before I got it, before it got released. So I think that the community doesn't really understand how that process works because I don't.
Yeah. There's bandwidth and resources. So for that reason, there's a workflow to drug discovery. But also, when does a discovery actually start? It's really iterative. And it's important to know that it's not like you have an idea in your lab and then you do an experiment overnight and then you have like, oh God, I got something that works. That, movie that it's great, but the Hulk did it, but that's not how it worked. So discoveries for drugs, it builds on, I would argue, decades before. There's two main areas of research. There's preclinical research and clinical research. So preclinical, it's usually pre-human. So we're thinking a disease, a model disease. We're thinking a transgenic mouse. We're thinking a petri dish where we have cells that are cultured, and we're modeling some aspects of the disease, and we're trying to understand the mechanisms that drive disease progression. So there's a lot of figuring out about what causes the disease, what kind of biochemistry is changed in the disease, and that's all preclinical. For example, I was working on Alzheimer's and I was looking at certain neurotransmitter systems, but I was using a modified fluorescent protein that's called green fluorescent protein that a marine biologist in, I think, the 60s or 70s found in deep ocean creatures. It built on tools that were discovered in the 60s, 70s, and then new techniques, new technologies that are far from a disease state, but then I was using to elucidate new mechanisms that drive Alzheimer's symptoms. So then it's a continuum, but then once you have a viable hit preclinical, so you cure the mouse in your lab, then you go to phase 1. So phase 1, you have some safety data from your animals. You want to make sure that the risks are as minimal as you can for your first brave humans. So phase 1, it's usually in healthy volunteers. You're going to administer it and have this first observation in a handful of people that it is well tolerated and that you're going to have these preliminary measures of what we call target engagement. Does it go where it's supposed to go? Does it bind to the receptor you think it should bind? Or does it have, depending on the mechanisms, we have different ways to see if it suggests that it does what it's supposed to do. Not curing, but it's at least interacting where it's supposed to go. Then phase 2 is a little bit more subject, or subjects is not a good word, I guess. It dehumanizes, but it's a few more participants. And it's usually participants that have the condition. In the case of our drugs for Parkinson's, we usually would have a phase 1 in healthy volunteers, look at the pharmacokinetics, so how long it lasts in the body, because that's our objective, is to make it last longer in the body with carbidopa, levodopa. And then the phase 2, we use Parkinson's disease patients with experience motor fluctuation. It's a smaller sample, so that allows us to have more dimensionality to our data. So for example, with the Crexont phase 2s, we would give the doses, we would look at NDS UPDRS, so how well the symptoms were managed over time, every 30 minutes. We'd also take blood samples to look at the levels of Levodopa. So then we have a lot of dimensionality to the information, how much Levodopa there is at that moment, how well the symptoms are controlled at that moment. And then we try to gather as much information per patient as possible to have a better understanding of how the drug works. But because it's only a few patients, things like full safety panels and convincing efficacy data, you can't really have. So for example, if you have a serious but infrequent adverse event in a cohort of 15, if it happens 1% of the time, your chances are you won't see it. So before it's approved, you need a phase 3. So where you're going to have a much larger sample of participants, and depending on the effect you're expecting and the potential for safety or adverse events. In the case of Crexont, we're talking about around 500 participants for the phase 3. And with much more participants, then you're more confident in your safety. And you can have more elaborate statistics to really be confident in your efficacy as well. You can have enough variability in your population to start asking questions like, is there a patient type that responds well? Is there a patient type that does not respond as well? So the phase 3 is, you're iteratively build confidence. That phase 3, you're then already confident that there's a good chance that this is going to work because you're going to invest a lot of money. And unfortunately, of course, there's a financial component to it. We're spending money on the clinical trial, then it needs to be pay for itself after when we sell the product. So yeah, phase 3 is a larger group, more power in statistics and efficacy and in safety. And that can take up to 5 years for completion.
So with Crexont, for example, how long did that whole process take?
Oh, that's a good question. That number of years, because I know I looked at data sets that were like from 2011 early on. Yeah. Neurotary was a process. It also happens in parallel.
Yeah.
So
So can you explain that? Because that's something
yeah,
that I learned too, which I think is really interesting. That a lot of us, we, and myself included, call Rytari 2.0 But it turns out they were actually in development at the same time.
Yeah. Yes. Yeah. So before we get them, we don't baptize them until they're approved, right? They don't get a full name. So they're investigational molecules first. And sweet little IPX06 was, 60. IPX066 is now known as Rytary. And that was, IPX was for Impacts. That was a company at the time that eventually got bought by Amneal that was testing these formulations. So it was the 66th iteration that gave promising data in vitro, then in animal models, and then that was going to be tested in humans. IPX203 Crexont was the 203rd, but they were screened. So we did a lot of in vitro screens to try to see what kind of beads release the best and everything. IPX066 happened slightly earlier in time, but its features, it's got different beads that release at different rates, and the idea was to have a more prolonged coverage. And a lot of that information you can get in vitro. And it was, it happened earlier on, and then it was sent to human trials. And then there's a question of bandwidth. It took a lot of time and resource investment just to bring eventually Rytary to the market. And that kept us then busy with the clinical trials and the launch. And IPX203 existed before Rytary was a thing. Once it reached the market, then we could go back to our other improvements. So IPX203 has a lot more different features. So it has this extended release polymer that is similar technology than Rytary, but it also has features that are meant to optimize in vivo. So like the mucoadhesive polymer, it's not the most intuitive feature. So the mucoadhesive polymer we have, if you open a dose of Crexont, you're going to see some powder, which is the immediate release component that acts quite quickly. And then you're going to see a lot of little beads, or we call them pellets. And these pellets have different layers. And the outermost is a pH-sensitive layer that protects it in the stomach. The immediate release component is going to start acting, and then it's going to stay intact. And when it leaves, the pH changes because your stomach is acidic, and then it leaves the stomach, enters the small intestine, and it becomes more neutral. When it becomes more neutral, it dissolves the outermost and activates the layer just under that, which is the mucoadhesive polymer. And that mucoadhesive polymer is going to increase the affinity of these pellets to the lining of the mucosa and slow it down. The reason why that's important is like Rytary, or we can think of the perfect extended release formulation. If it was just extended release, but it's perfect, it would start releasing, which is wonderful. Then it would travel in your small intestine, and it would keep on releasing, which is again wonderful. But eventually, it would leave the proximal section of the small intestine, which is where the absorption of levodopa. Levodopa is an amino acid, and it relies on what we say active transport. It needs to be transported by a transmembrane protein across the lumen to make it from the gut to the systemic circulation and eventually the brain. So it has a window of opportunity when it's released to make it to systemic circulation. So even that perfect bead that magically releases for the full time of transit, once it's downstream of the jejunum, which is the part of the proximal small intestine, it doesn't work anymore, and your levels are going to start going down. So IPX203 has this mucoadhesive polymer, so it increases, at least in theory, the time that it will spend at that site. And it's reflected in the data. We don't have an ultimate demonstration that it attaches right where it's supposed to, but it lasts longer than Rytary in humans. And the bioavailability is higher. So bioavailability is like how much you put in the system, how much the dose is, and how much the body's going to be exposed to it. And you always lose a little bit of drug with the oral formulation. And you lose more of the levodopa with Rytary than with Crexont. So that's an indirect suggestion that it actually does what it's supposed to do and increase that window of opportunity that even would limit even the best extended release formulation. But that can only be tested, it cannot be tested in a dish because it doesn't have an intestinal lining, it doesn't have all these features. So IPX203 could not be as quickly of a okay, let's go and give our better formulation. It required more testing before it was as promising as we know it can be. Yeah, it depends how far in the future we're looking at, right? So there's many different avenues and there's many different limitations, but that's a great question though. I'm just trying to figure out a way to answer it in a reasonable way.
Right.
So, in terms of levodopa, I think in the grand scheme of things, we want more disease modification. We want a cure. But in terms of symptomatic management, we know that levodopa, if it's just right, it still works really well, even in the context of motor fluctuations. It's got to do with how much is in your body and how well it's absorbed and all these challenges. So, the subcutaneous infusions are an advantage over the intraduodenal, like tubes. They don't require a minor surgery, but then they have nodules, right? And you're also limited in the total amount of levodopa you can give in a day. But, changing that formulation to make it more compatible with continuous subcutaneous. So there is likely to be strides in the formulation there to make it more compatible with and tolerable by the skin and the immune system. In terms of oral formulation, we are now thinking about prodrug. So you could release a prodrug that would accumulate in situ in the body, but then would slowly be converted to actual levodopa. And the prodrug could be protected from degradation. So there's all these different avenues. You could think, levodopa is an amino acid. You can think of other vehicles that would slowly deliver levodopa. But levodopa works great for symptomatic management. But if we want to be ambitious, I also think that there's gene therapies. Right now, there's GDNF trials that are pretty promising. So GDNF is glial-derived neurotrophic factor, and it's injected in the brain through a viral vector. So they engineer a virus, they trick it so that they replace the virus's genome with the sequence for this neurotrophin, and then it's been injected now in few humans, and it releases GDNF, and it seems like it's having pretty promising effects. So neurotrophin, genetic engineering, there's so many avenues, and it's important to keep investing in all of those because they're all going to yield at different times with different promises.
Can I just say how this man's face lights up when he talks about mucosal lining and polymers and stuff? Like, I'm not going to call you.
You can.
Talking about those pellets, like those were his children. He's talking like I talk about Sephora. One of the big takeaways I want to have from this particular episode is that everybody that I've met in pharma has been people that have love and passion for what they're doing, and that changed so much of my thinking. And this is kind of how, first of all, you mentioned you came from Alzheimer's research, and what I often find is like people have connections to other neurodegenerative diseases, people in their family who've had these conditions, and that's, part of their origin story too. And it makes, I always say, like love is so important in life and in Parkinson's. And I see the love that you have for science, and that's a really beautiful thing. But I also say that to me, I would feel like a hypocrite to dump on pharma because I get up every morning and I take a pill. I mean, that's the reality of my life. And I am going to get off this call and take another pill. And
that's where we're heading.
That's what I need to live my life, to take my kids to school and spend quality time with my family. So I rail a little bit against the big bad pharma narrative because of this, because the fact that, we're here and we're learning from each other, not just us as individuals, but like that's how the process works. And that realization and is what I think that we need to build on, and because I'm not going to cook up the cure to Parkinson's in my kitchen, but we have to partner, and our doctors have to partner, and we all have something to give and to learn from each other. And if nothing else, also, I want to encourage people to participate in research because, like he was saying, the GDNF trial is a perfect example. You had DBS. Somebody had to trial. Somebody had to sacrifice their bodies for us to get here. And those are the real heroes to me, you know? And so I really, really encourage people to, sign up and participate in research and ask their doctors if they're aware of any studies in their areas. Go to clinicaltrials.gov. Michael J. Fox Foundation has a great portal for that as well. And, because we need all hands on deck. We're all trying to get to the cure faster, but how many people are signing up for these trials, And that can be the little part that we play too. I want to touch on really quickly, though, some legit beef that I have with the preclinical model, which is something that I heard from my friend, the advocate, Rochelle Flanagan from Northern Ireland, which is that maybe you can touch a little bit on the history of this, but that, most of these drugs were not trialed on women.
Oh, yeah.
And that they actually don't even use female mice in the preclinical model. And that we also were talking earlier about young onset and the lack of representation of young onset in those trials as well.
Absolutely.
So can you speak to that a little bit?
Yes. So there's different layers to that question. But so in terms of preclinical, it's true that at first, like we're talking, when I started doing research, in fact, when I was writing my grants, you have to justify how many animals we're using, and for money, which is a bad excuse, and also reduce animal suffering, we will chose to go only for a homogeneous male rat sample. But it's silly to think about
the female rats don't even have a seat at the table, y'all.
Right. But then right
because they didn't want to pay more money for female rats. That was fun.
But it's changed now, so that's good. So the more recent grants that I wrote, actually, in fact, if not using equal amounts of males, and but for the record, not that I want to expunge myself, but I was using a balanced population. But when writing grants, we had to justify. So there's some evolution. We had to justify that we were using both male and female. So but we're talking, we're for silly reasons like fewer animals or sparing mice. But what about the human? Like you want to use half the amount of mice, but like how about the half the amount of humans that are not going to be represented in that trial? So that argument makes sense. So you're completely right, but there's direction in progress. Is it fast enough? Probably not. But there's a level of awareness, but it's important to keep having these discussions. Now, in clinical trials, it's really interesting, where it's then again, women were underrepresented, getting better. When I was analyzing our clinical trial, I was happy to see that it was much closer to parity. So that was good. But we also think of, as yesterday we met at a different conversation, and racial disparities and underrepresentation, and for example, African Americans are on average not followed by the same kind of clinicians, are not seen at the same centers, are not aware of the clinical trials as much. There's also a confidence that we need to work towards historical reasons and many valid reasons. So there's a lot of room for progress. And then again, advocacy and discussions. I think, pharma opening up conversations with clinicians is important, because if we want to be accountable, if we want to build trust, we need to have our clinician collaborators and have transparency and work with them. Same thing with the patient, communication, advocacy. If we want to build trust and the respect, then we need to have open conversations and it's a live process. It's not having one seminar here and then we're done. It's to have these lifelines and to keep that open. So any effort into opening channels of communication is extremely worthy, in my opinion. So lots of room for progress, and you're totally right. Your beef is justified. And I hope that addresses your point a little bit. But technology has goods and bads, but usually it tends to bring, in a good direction. I think wearables are going to help a lot to generate continuous data. Because, for example, bring it to our clinical trials because this is what I understand the most. But our first step in our clinical trial was just to optimize the drugs that the patients were already on. Because all these patients were seeing neurologists, and I would assume that they were all qualified and good. But once they entered the clinical trial, they started filling what we call them Hauser diaries. So every 30 minutes, they're saying, am I on? Am I off? Am I experiencing dyskinesia? They're generating this data. And then they're coming to their appointment, part of the clinical trial. They're feeding this information to the clinician. Now the clinician has quantitative ways to be like, oh, you turned off predictively here and there. Let me shorten that interval. And that information, we saw a significant improvement. They're on the same drug, but then now they're part of the clinical trial. They're providing that information, and they're doing better, significantly better. Not as much as when they changed formulation, but it was non-negligible. And it's the same drug. So I don't think the solution is to get everyone to fill Hauser diaries all the time because the burden is unreasonable. But wearables and how we can decode Parkinson's is complicated because how you turn off is different than how Chris turns off. So we need more fancy algorithmics to do some good deep learning on that. But we're getting there. The algorithmics are getting there. The wearable technology is getting there where patients could, without increasing their burden, which is already up there, have more rich data sets to help the clinician. So this is, I think it's a hopeful direction. And I think technology is going to be really useful there to, the more data clinicians have, the better equipped they are to adjust your medication. So it's about breakthroughs, but it's also about using the existing medication as appropriately as possible, especially in the case of Parkinson's, where one Parkinson's disease patient is one Parkinson. The adjustments that need to be done for the proper drug to work are quite impressive. It's a very good point.
So I'm on Parkinson, and so I keep bringing it up because it's all about me.
It should be.
And I have a question because, of my own experience with the dosing.
Mm-hmm.
All right.
Mm-hmm.
So what goes into how the dosing tables that rule our lives are developed?
Oh, yes. And that's going to bridge into other points that we talked about. So that's one of the many reasons I love your question. So clinical trials, pros and cons, right? There's not really excuse, but for example, the Crexon trial, we had a certain variability in our population, but part of the inclusion criteria was that the patients were not on Rytary, they were not coming on CR as the main dose, they were not taking COMT inhibitors like entacapone or picapone. So because we had to test the conversion and we had to have enough power to test that conversion, they were like, we're going to find a group that's the most frequent, test it, and we based ourselves on pharmacokinetics. So we gave IR to patients in phase 2, then we gave Crexon, and we tried to find conversions that would match. But Levodopa, the pharmacokinetics are notoriously variable. So then in phase 3, based on our understanding of pharmacokinetics, we're testing an algorithm in a selected most likely population, which means IR not taking other medication. And that worked for the majority, and we saw it in our phase 3. But then to go back to how to live science, MSL, we have our early adopters. They're starting to prescribe it. And that's an example. We're going to present it at an NDS, and I'm excited for that. So I can't give too much details because it's not published yet. But we went out in the field. Of the questions we're asking is, how is the dosing going? Do you have to do a lot of adjustment? This is what we can anticipate from our clinical trials. But then again, it's an experiment. We have inclusion, exclusion criteria. This is not the real world. So what can you tell me about different patient types? And now we're gathering that intel about conversions from Rytary, about conversions in people with familial, early onset, and we bring it back. And we actually already have a success story with that. So we heard frequently from our early adopters that the algorithm, so the table that we tested in the clinical trial, they said, yes, like your data, it works for the majority of patients. But we have a non-negligible minority, the key word in general, is that when we convert them, we dose them way too high. It's way too strong. They're not doing well. Some of them, it was strong to a point where they took their first dose, did not want to hear anything else about Crexont, wanted to go back to IR because the experience was borderline traumatic, if not fully traumatic. So we learned that, which is but then conversation is really important and the access is really important. And then I went back into clinical data and I looked for a signature of what they reported. And in fact, we found it. So we found certain patient types that were dosed way too high, and using the data and what we call post-hoc analyses based on feedback, we could now generate new quantitative data that can help the clinician find these patients at higher risk of being overdosed. And then with all this new data that is generated after approval, now they can be in a better position to minimize how many patients are going to be dosed inappropriate. So it's really a dynamic conversation.
And it's ongoing, right?
It's ongoing.
I mean, it's not like, okay, that's that. It was it.
Move on.
And that, I think, so important because I feel that is what happened, like when I started I wasn't dosed correctly. And I always go back to these shared roles, these shared responsibilities, and that we have to feel empowered as patients to not wait and give feedback to our doctors, good, bad, or indifferent, so that they can kick it up the food chain and then we can get our doses adjusted too. I mean, you guys know it's like fine line. And, especially since I have dyskinesia and stuff, like I know a lot of people suffer needlessly because they're just not good getters with their doctor. And it's time consuming, especially if you get follow-up appointments and dealing with nurses through the portal and calling the office and all that stuff. But I think that the dosing is so, so very important. And we have to just keep searching for our sweet spot. And of course, it's a moving target, right? I mean, we're chasing a moving target. But I mean, try and keep giving stuff a try, whatever it is. I mean, it's not specific to any one thing. I mean, you guys know. I've tried Mucuna. I've tried Rytari. I tried immediate release. Like, I'm an equal opportunity drug trier because I just want to see what combination works for me. But that's like one of my pet peeves is when I see people that just are not willing to try new things. I understand the fear, but it comes with the disease. It's scary. But if it doesn't work, you can always try something else, no matter what the product, And that's been my experience too,
Yeah, because if it works, you don't mess with it. But there's a progression of disease that you got to try something eventually because it's either not one of the others. It's going to happen.
Bridging that to your point of the adjustment, and it takes work from the patient and the clinician. And that's one of the reasons why it's so important to advocate for yourself, because clinicians, neurologists are overwhelmed. They have a finite amount of time to spend with the patient and they need to prioritize. And changing medication is definitely worth it, and trying, because, in the end, you want the best possible management as possible. But these overwhelmed neurologists, if there's no interest, or I'm not victim blaming it, but if there's no, it's important to advocate for yourself, because if there's no interest from the patient, they're much less likely to volunteer a lot of their bandwidth. If the feeling is that the patient is satisfied with their symptomatic management, why would they try to adjust, do all these adjustments and sacrifice some of their bandwidth for that? So it's really important to communicate.
And then you get like really not troublesome, interesting patients that just ask all the whys, like, why does this, why is, too passionate? So I have experiencing anxiety related or anxiety just dystonia. And act by yes to it. So is it Parkinson's, or is it just an anxiety this was presenting? That's
and
she gave me
that's interesting. And so I'll show that there's something that's referred to as functional movement disorders. And those are not mutually exclusive with, your typical, they're all physiological at some point. There's a negative connotation to functional. Some patients, sometimes they hear it's functional, they feel like it's not valid. It's extremely valid. The behavior also happens with, if anything, more complex neurophysiology in your brain, physiological or as real. It's just more complex neuroscientifically speaking. So, but also you can have a non-functional disorder like Parkinson, then levodopa is one of your helpers. But that can actually, that's the brain is a complex organ with this exquisite circuitry. So if you have a non-functional disorder that is triggered by a neurochemical imbalance, well, it's likely that over the years, that's going to start affecting your circuits, and now they talk to each other and increases the likelihood that you're going to have also a functional component to your disorder.
Yeah.
But it's as valid if the solution is psychotherapy. I think there's also like structural. The neurologist is really important and they're really overwhelmed. But, you think about centers, like I think of Innova, that they have this very multiple, they're not alone. I don't want to skip on important people, but for the sake of conciseness, occupational therapy is part of it, physiotherapy is part of it, psychotherapy should be part of it. And also because the neurologist cannot answer all the questions first because they're not everything. And also their bandwidth. So if some of the explanations can be relayed to an advanced practitioner, that's great, But then again, not all centers are equal. Some of them have more resources and different patients have access to different. So the quality of care really varies based on many factors.
Where you're at.
I would say that geography and luck and access and, obviously our financial consideration are the real drivers behind the care that we get, what drugs that we get, and all of that good stuff. But as much as I think the pharmaceuticals, we always talk about mindset and exercise and support and all of those other components that need to kind of chase a more holistic aspect. And me, like the pharmaceutical is just like that piece of that puzzle, a very, very important piece, but it's not the whole. My trainer always tells me, you take medication so you can exercise. And that is true. I live and die by that. This is just one piece of that pie. It's a tool in the toolbox. Exactly.
Yeah. But they're connected. The teams are connected. So I was talking to KOL that is really into exercise and high intensity. And he was telling me that levodopa is not disease modifying. We all know that, but exercises and having proper symptomatic management, having your best formulation, your best adjustments, having the best cocktail that works for you allows you to be more active, to exercise more. And exercise is disease modifying. So, we shouldn't frown on, silly symptomatic management if it allows you to have first there's the compassion, right? But also it modifies your lifestyle.
Well, thank you so much for your time, Simon.
My pleasure. Thanks for having me.
You. I forgot to put it on this one.
Very much.
All right. Bye.
Thanks,
Chris.
Thanks for tuning in to the Living Well Starts Here vodcast with Young Onset Parkinson's Network.